Hello everyone. We are gonna talk about neurotransmitter synthesis today. We are Efrain, Fatima, and Kayleen, and we'll focus specifically on biogenic amines, acetylcholine, and peptide neurotransmitters, which we think are the most important neurotransmitters because they're whole different neurotransmitters, and we chose the most the key ones. We'll break down the enzymes and the reactions involved so you can understand not just what these molecules are, but how neurons produce them. So, first of all, what are neurotransmitters? Neurotransmitters are the chemical messengers that neurons use to communicate. They are synthesized inside the neuron, then stored in vesicles, and finally they are released when calcium enters the presynaptic terminal. For this, there are three major classes which we are going to talk about: amino acid neurotransmitters, biogenic amines, and peptide neurotransmitters. Each one has different functions and different synthesis pathways, which we'll explain in detail. So, first, how are they made? How are these neurotransmitters made? Each neurotransmitter has a different um, pathway, but mainly for the three of them, we have to think about three main components for all synthesis of neurotransmitter. We have precursors, which are usually amino acids. In this case, we're going to talk about amino acids or small molecules absorbed from the diet. Then we have the enzymes inside the neurons. These convert the precursor step by step into the final neurotransmitter. It's like all the previous pathway we've seen of lipids and carbs. We use enzymes to make those reactions. Well, it's the same in this case of the neurotransmitters. And finally, the vesicles for the storage. So once synthesized, the neurotransmitter is packed into vesicles so it is protected and ready for release. Basically, this happens in the neurons. It cannot go out just like that. So it's like it's inside the vesicles, which are like their protection, their their way to travel from neuron to neuron. And each pathway is carefully regulated, and in several cases, the availability of the precursor or the activity of the rate limited enzyme determines how much neurotransmitters are produced. Well, first, I'm going to talk about biogenic amines. So, you must be asking, what are biogenic amines? These are neurotransmitters derived from modified amino acids. There are different types. The main ones are catecholamines, serotonin, which we know very well, and histamine. We're going to talk about these three, the synthesis of these three. All of them are crucial for mood, for alertness, autonomic responses, and even endocrine control. What they have in common is that their synthesis starts with an amino acid and requires very specific enzymes. So, well, next we're going to explain each synthesis pathway in detail, starting with catecholamines.
Catecholamines. They include dopamine, norepinephrine, and epinephrine. All three are synthesized from the amino acid tyrosine, and each reaction uses a different enzyme. Here's what happens step by step. First, we have tyrosine, which goes to L-DOPA. The enzyme it uses is a tyrosine hydroxylase. What this enzyme does is it adds an hydroxyl group to the tyrosine. Its importance is that it is a rate limiting step, meaning that it controls how much dopamine, norepinephrine, or epinephrine the neuron can make. Then the L-DOPA transforms to dopamine. The enzyme it's used is the DOPA decarboxylase. What it does is it removes a carboxyl group, and this reaction is very fast and requires vitamin B6. Then the dopamine goes to norepinephrine. The enzyme is the dopamine beta hydroxylase, and it adds a hydroxyl group to the dopamine, and this occurs inside the vesicles. Then the norepinephrine transforms into epinephrine. The enzyme used is the phenylethanolamine and methyltransferase. This transfers a methyl group from SAM to NE, and this mostly happens in the adrenal medulla. Now let's talk about functions. The function of the dopamine is to reward movement and motivation. The functions of the norepinephrine is alertness, attention, and sympathetic responses. Then the epinephrine function is to respond to stress, and it has cardiovascular effects. Serotonin is synthesized from the amino acid tryptophan, and the steps are the tryptophan goes into a 5-hydroxytryptophan. The enzyme is the tryptophan hydroxylase. What it does is it adds an hydroxyl group, and this is the rate limiting step for the serotonin. Then the 5-hydroxytryptophan goes into a serotonin, and the enzyme is an aromatic L amino acid decarboxylase, and what it does is it removes a CO2. The functions of the serotonin is that it regulates mood and emotional stability. It controls the sleep and wake cycles. It also modulates the appetite and helps regulate gastrointestinal motility. Histamine comes from the amino acid histidine. The enzyme it uses is the histidine decarboxylase. What it does is it removes the carboxyl group, forming a histamine. It requires vitamin B6 as a cofactor, and the functions of the histamine is it regulates inflammation and immune responses. It also stimulates gastric acid secretion and maintains wakefulness and attention in the brain.
Now we're going to talk about acetylcholine. It is unique because it doesn't come from an amino acid, and it has several essential functions, such as at the neuromuscular junction, it triggers skeletal muscle contraction. In the parasympathetic system, it slows heart rate, increases digestion, increases secretions, and causes bronchoconstriction. In the central nervous system, it participates in attention, learning, and memory. Acetylcholine is synthesized from acetyl-CoA and choline. With the help of an enzyme called choline acetyltransferase, it transfers an acetyl group from acetyl-CoA onto the choline, producing acetylcholine and releasing a CoA. This is a rate-limiting step. The reuptake of choline into the neuron. If choline isn't transported back efficiently, the neuron cannot make enough acetylcholine. Peptide neurotransmitters. They include molecules like substance P, endorphins, and enkephalins. These are very different from small molecules neurotransmitters. Instead of being synthesized locally in the terminal, they begin as large precursors proteins made in the neuron soma. How they are made. A gene is transcribed into mRNA. The cell produces a large protein in the rough endoplasmic reticulum. The protein is processed and cut in the Golgi apparatus. Then the active peptide neurotransmitter is packed into vesicles, and those vesicles travel down the axon to the terminal. We have some functions such as pain modulation for substance P, stress response and analgesia for endorphins and enkephalins, emotional behavior, and autonomic regulation. Because they require gene expression and protein processing, peptide neurotransmitters are slower to produce and not rapidly replenished, like small molecules neurotransmitters. To summarize, neurotransmitter synthesis depends on three main factors: the precursor available, the enzymes that modify those precursors, and the storage mechanisms that prepare the neurotransmitter for release. Each pathway is tightly regulated and essential for normal brain and body function. Thank you for your attention.